Hello, students. Welcome to the session Biology Grade 9. Today, we are going to discuss about the discovery of cell. Today, we will go back in time where scientists made different types of discoveries. So let's start together, okay? Before we dive into the journey, I just want to have a look on learning targets. Understand how cells were discovered. Know the key scientists behind the discovery. We will discuss about the different scientists. And after that, we will observe and identify cells in an onion peel under a microscope and develop curiosity about cell structure. Are you ready, students? Okay, fine. Now I'm going to meet you the scientist who discovered cells, who discovered cells in a very beautiful way. That is Robert Hooke. He was the scientist who discovered cell in 1665 using a primitive microscope. This is the microscope which is made by the Robert. And he has taken a slice of cork, the bark of the tree, and he placed it under the microscope. This was the structure under the microscope of Robert Hooke. You will think that this is not fancy microscope. Yes, but it was good enough. It was powerful enough to see a very thin slice of cork. What he observed inside the cell, inside this structure, that the thin slice of cork is made up of small compartments. These structures reminded him of small rooms in a monastery. After that, he named them as cells. The word cell came from the Latin word cellula, which means little room. So this is how the cells were discovered. But this scientist discovered cell that is non-living cell because cork, it is a bark of a tree and these cells are non-living. Now let's meet another scientist. Okay, fine. Students, Robert Hooke discovered cell, but biology didn't stop there. I want you to introduce to some superheroes of cell biology which covered all the secrets, which uncovered all the secrets of cell. Let's start with the very first scientist, Antony von Leeuwenhoek. He was the scientist who discovered protozoa and bacteria. Antony von Leeuwenhoek, he was a Dutch scientist. He wasn't rich and not famous. Actually, he was a cloth merchant. He made a powerful lens and observed different types of microorganism. Actually, he placed drops of pond water under the microscope. There, he will found that inside the pond water, there are different minute microorganisms are there, which are floating under our microscope, which are moving under the pond water. From there, he named that animal as animal cues. Okay, I will discuss about this in the next slide. Next, after Leeuwenhoek, next scientist came Robert Brown. He was the scientist who discovered nucleus. Robert Brown was studying plant cell and found that inside the plant cells there are dense, dark colored area. And then he named it as nucleus. Now let's meet two other scientists, Schleiden and Sean. Schleiden, he was a botanist, means he was studying on plant cells. And Sean, he was a zoologist. He was studying on animal cell. Both were studying on different organism and they teamed up and said, wait a minute, plants and animals both are made up of cells. So that was the very first postulate of the cell theory. So these scientists, they founded the cell theory and they have given the number one statement of the cell theory that every living organism, plants and animals, they are made up of cells. Last, Purkinje. He was the scientist who coined the term protoplasm. Although protoplasm was discovered by Dujardin, but that time he named it as sarcode. It was a kind of jelly-like substance which is present inside the cell. After that, Purkinje was the one who named it as protoplasm. So protoplasm was discovered by Dujardin and it is named by Purkinje. Good to go? I just want to show the discovery of Antony von Leeuwenhoek. He was the person who discovered living cells. Okay, so here you see this was the lens which was made by the Antony von Leeuwenhoek. This is the picture of Antony von Leeuwenhoek, and this is the picture of the discovery of Antony von Leeuwenhoek inside the pond water. And it contains white blood cells, red blood cells, moving particles like bacteria and sperm cells. So just because these particles are moving inside the body, he called them as animal cues. He named it as animal cues because movement is one of the property of animal. So just because these particles are moving, and he named it animal cues because these are very tiny organisms. These are microorganisms. So little animals like structure. Next, move for next slide. Sorry, students, for the delay. Now, this is the picture of Robert Brown, scientist who discovered nucleus. And when he was actually studying the plant cell, after observing nucleus, he has given the statement observo el nucleo di la cellula. It is a kind of Latin language, okay, which means that cells inside the cell, there are dense, dark colored structures are there that are nucleus. So this was the picture taken by the Robert Brown. Next, Purkinje. Purkinje, I told you that he coined the term protoplasm. Students, what is protoplasm? Protoplasm is the living part of the cell. It means that it contains cytoplasm and nucleus. So yes, if in any cell, cytoplasm is present and nucleus is present. So they are collectively called as protoplasm, and yes, that cell is living. So protoplasm is basically the living component of the cell or the living part of the cell. It is the collection of cytoplasm plus nucleus. Now, students, it's time to do some lab activity. Okay, it's not just activity, you just have to look through the microscope, but it's like you have to step into the shoes of Robert Hooke and see what he saw in the year 1665. This is the activity 5.1 from NCERT. To observe and identify cells in an onion peel under a microscope. Yes, we are going to discuss the cells inside the onion peel. Yes, onion, the one in your kitchen, is now going to become a hero. Let's start it with the activity. These are the step-by-step process. You have to break it together, and I want all of you to imagine it doing. Start. First, you need materials. Of course, if you want to prepare something or if you want to cook something in your kitchen, you need materials or ingredients. So for preparation of the onion slide, you need materials. These are the materials. You need onion. Students, what do you think why we are taking only onion? Any idea? Why we are not taking any other kind of apple peel or leaf peel? Any idea? Go through this. Just think, have you ever chopped onion in your kitchen? Have you ever cried while cutting onions? Yes. If you cut the onions, you will find there is a thin layer inside the onion. Inner side of the onion contains a very thin transparent layer, and this transparent colorless layer is powerful enough for microscopic slides. So this is the reason why we have taken onion. Next, you need transparent cover slip. Saffron, it is a kind of dye. You can also use iodine or methylene blue as well. Forceps, dropper, microscope, and of course slides. So let's do this activity together. Are you ready? Students, you can perform this activity in your lab with your teacher. You can discuss more with your teacher and perform this activity. But now it's time to listen how we have to do this activity. This is the step-by-step procedure. First, pull inner layer off. Students, what will you do? Take an onion and chop it into in pieces. Take one piece and pull off the inner layer of the onion that is transparent with the help of forceps. Now, next procedure is place the thin layer at the center of the slide. And yes, while placing it, make sure that this layer should be flat. It should not be folded. Otherwise, you will see traffic jam, cells traffic jam under the microscope. Next procedure. Now you have to add 2 drops of iodine or methylene blue or saffron like stains, just like here in this picture. Okay, and leave them for a minute. Clear? Next, what? This is the last procedure that you have to do. That placing a cover slip on the slide. See, while placing cover slip, make sure that the cover slip should be placed gently. Otherwise, air bubbles will come when you look under the microscope. So this is one of the most important procedure while placing the cover slip. Okay. So with the help of needle, you can place it gently. Now it's time to look magic. Okay. It's time to show magic. I want to show you how it will look under the microscope. Look, students, this is the observation. When you look under the microscope, you will see break-like structures. Amazing, is it? And this is colored. Why? Because we have used dyes. We are using stains or dyes to make the cellular structure visible because inside the cells, the structures are transparent. They are very small. So this is the reason why we are using colors. We are adding stains to make them properly visible. So inside the microscope, you will see that these are the structures present in the cell. Onion cells, brick-like structure, they have a cell wall which is rigid. Inside the cell, there is a dense nucleus, is there, dark area. And this is the area where different organelles are present, cytoplasm, and ultimately a large vacuole is present. We will discuss these structures later in this chapter. Now here comes the conclusion of the activity. What we have studied, what we observed, that onion cells shows typical features of plant cell, because onion is a plant, and it shows typical features of plant cell. That is, it shows present of cell wall, large vacuole. Microscopes are essential to study cell structure, of course. And last, plant cells have a rigid cell wall and defined structure. Cool. That was all about the concept and activity. I'm sure that you got clear. Now, students, it's your time to have a recap of this class, okay? And I'm just giving you 2 to 3 minutes before we will start our activity-based questions, okay? Just read it again, whatever we have studied. If you have any doubt, you can ask your teacher in the class. Take your time, go through, go thoroughly. Okay, what are the topics we have discussed? I'm sure that you have done with the revision. Okay, student, now it's time to do some questions. I want to check all of you that whether you have grasped it or not, what we have studied in the class. Okay, it's time to do some questions based on the previous topic. Multiple choice questions. The very first question is, what year was cells first discovered? These are your options. And I'm giving you students 10 seconds to answer this question. Tick, tick one, tick, tick two. Do it fast, very easy. Don't shout in the class, okay? Because your teacher is also there. And the answer is, what is the answer? Yes, it's C, 1665. Let's move to the other question. What did Robert Hooke observe to discover cell? Onion peel or cock slice or pond water or bread mold? Tell me the correct answer. You have 10 seconds. Are you ready? Okay, so your answer is option B. That is slice of cork or cork slice. Next question. What does the word cell mean in Latin? I told you, the very special term I used. Your options are room, life, seed, and micro. What is the correct answer? Yes, it is option number A. That is room. I given you the word cell means it came from the Latin word cellula, which means small room. Next, what instrument helped in the discovery of the cell? Telescope, periscope, microscope, or spectroscope? Tell me the correct answer. Very simple, students. You have studied only one instrument here. Simple. Option C, microscope. Good to go. Another question. What did the saffron stain say to the onion peel? Let me bring out your true colors. I'm here to die for you. Just a drop, and you'll be famous. Time for your close-up under the scope. Tell me the correct answer. I'm sure you are two are confused in option A and B. And let me bring out your true colors or I'm here to die for you. What is the correct answer? I'm here to die for you. Stains are used to dye the cellular components. Now, rapid fire question. Students, you have to answer fast, okay? Your very first question, who coined the term cell? Who is going to give the answer first? Who coined the term cell? Yes, it was Robert Hooke. Question number two. What did Leeuwenhoek observe in pond water? You have only 2 seconds. Simple, animal cues. Next question. Again, a mystery question which we have discussed yesterday. Okay, there's another mystery in front of you. Look at the statements. I'm shaped like a room and have no door. Hooke named me while he looked at Cork's core. Though I look empty, I hold life spark. What am I that he saw in the dark? What he saw inside the microscope when he was actually studying the slice of cork. Small room-like structures. Okay, so what are the structure? A cell in the cork. Clear? Next, I saw things in water that danced all around. Tiny creatures that made not a sound. Animalcules. I call them with pride. Who am I with microscope by my side? Your clue is given here. Students, answer it fast. Simple. Who has given the term animalcules? Anthony von Leeuwenhoek. Clear? Next, now it's time to have some discussion. Your question number one, what helps maintain the shape of onion cell? When I was showing you the picture of the observation under the microscope, I told you which structures is going to give the proper shape to the onion cell. Of course, it is a cell wall. Why is a stain used while preparing the slide? Why we are staining the slide? Why we are staining the onion peel? Mhm. To make the cellular components visible, because cellular components like nucleus, cytoplasm, they're transparent. They are very small. Okay. So we stain them so that they will be clearly visible under the microscope. Next, it's time to take a pause and reflect. If microscopes weren't invented, what would we still not know? Can you imagine, students? Mhm. If microscopes weren't there, we would not able to see the cellular structure. What about bacteria and viruses? Would biology and medicines be the same? No, of course. So this is the reason why if microscopes weren't invented, the mystery remains unresolved. Okay. Because nowadays we are able to see different types of microorganisms and microstructures inside the microscope. And that time, if the microscopes weren't there, it would not possible for us to look at these minute structures. Next, why do you think cells remained invisible for so long? Simple, because that time microscopes were not there. Clear? So that was all about the session, students, and these are your takeaways. Every great structures begin with a single block. Okay, say it with me. Every great structures begin with a single block, and every life begins with a single cell. Okay, students, that's all from my side. And if you have any queries, you can ask to your teacher. Okay, and keep exploring the tiny structures and keep doing experiment to study more about these structures. Thank you.